So I'm going to talk about clinical trials now and in the future. And I'm really honoured to be presented with the David Grahame Smith Prize. He was one of the UK's leading clinical pharmacologists. He was famed for his work on serotonin and carcinoid syndrome. Unfortunately, he retired the year that I began my clinical pharmacology training, and I don't think I ever met him in person. His extracurricular interests, according to this article by Jeff Aronson, were wide and often surprising. And one thing that stood out from this article was he was a vigorous player of water polo, a game in which there is fair play above the surface and dirty work below, the perfect training for an Oxford professor. So whether that's true or not, I don't know. But in Dundee, I think we try and do fair play most of the time. So it made me reflect briefly on my career and how I came to become a clinical pharmacologist. I was interested in medicines and what they did for people even before I went to university to study medicine. I had a Saturday job in a chemist shop. I did some school projects on analysing medications for the amount of iron in them. And I think I was inspired at Aberdeen, which had a very strong clinical pharmacology unit, winning a Seneca Pharma Prize in my third year for clinical pharmacology. I had the privilege of spending a fourth-year summer project in the labs of Ben Benjamin, looking at nitric oxide in the stomach. And I went on to work in the clinical pharmacology house job with people such as Jim Petrie, John Webster, James Macleay, and Mary Joan MacLeod, all of whom were excellent physicians and had a real interest in driving drug therapy forwards. I moved on to Edinburgh, training with various people, Paul Padfield, who was a great supporter of evidence-based medicine, Ian Wilkinson, Stephen Waring, who were registrars at the time, Simon Maxwell, David Webb. And it was Ian who encouraged me to apply for a post in Cambridge that came up as clinical lecturer, where I was again privileged to work with Maurice Brown and Ian Wilkinson and completed a PhD in clinical pharmacology. I moved to Dundee in 2008. It was my first consultant job, and I came to work with Tom, who had just started off a large streamlined clinical trial, and I've been there ever since. And I'd just like to take this opportunity really to thank Tom. He's taught me a lot over the last 10 or 11 years about clinical pharmacology, and he also came in very handy occasionally as a babysitter during his lunchtime nap. This was a few years ago now. So I work in Memo Research in Dundee, and our aim is to improve the safety and effectiveness of medicines. We have 3 academic leads. Tom is the director, I'm the deputy, and we also have Alex Doney, another clinical pharmacologist who joined us about 3 or 4 years ago. And we run a team of about 50 staff members working on various studies there. So our interest is large-scale randomised clinical trials, and more recently getting into decentralisation of clinical trials, which I'll speak more about later, and we also do some pharmacoepidemiology work. And we currently have about 40,000 participants taking part in our clinical trials. So we're a busy unit, we don't have any downtime really, but we enjoy what we do. So traditionally, clinical trials worked by recruiting people via local clinicians. There were vast networks of local investigators. It usually involved multiple clinic visits for patients attending central sites, often in hospital settings. There was limited flexibility in the timing of the visits for patients. There were large numbers of staff involved. In the past, there was little patient involvement in either the design or the delivery of the trial. And there was little feedback to patients of their own individual results at the end of the trial. Generally expensive, slow, and clunky. And we still do things this way in many places, and many trials are done this way. But how can things be made better for both the participants and the people running the trials? So we've always had an interest in getting the public involved in research, and we ran the Get Randomised campaign some years ago that was published in the BJCP at the time. And this was trying to promote the idea that clinical research was important and to try and get the public interested in taking part in research. And since then, we continue to have patient and public involvement in our studies. So we have input into the study design and processes. They help us with preparing and revising study documentation. We have patient or lay members of trial steering committees. And we've recently set up a more formal public involvement in Memo Studies group so we can get advice on various aspects of running trials. We've also been interested in using technology to its best in trials, and again, this was a few years ago now, but the swine flu vaccine, when it came out, we rapidly set up an online study to try and follow people up who'd had the swine flu vaccine and look for side effects. And the best thing about this was we had a large number of people who had no side effects who could report that they felt fine after it as well. And we managed to publish this, and it was used as part of the evidence by the MHRA at the time for the safety of swine flu vaccine, including in a cohort of pregnant women. One of the big trials we did in a more streamlined way was the SCOT trial. This is a multi-centre streamlined trial of cardiovascular safety of celecoxib versus traditional non-steroidals in patients with arthritis. And this trial ran in the UK, Denmark and the Netherlands with more than 7,000 patients randomised looking at cardiovascular, GI and renal outcomes. And we tried to make this more streamlined, firstly by getting the medication just prescribed by their own normal primary care physician. And we followed them up for events using record linkage to national databases where the events are collected from NHS data or the equivalent abroad. And this gives us information on hospitalisations and deaths. So some of the advantages of using these centralised databases for events, you get more complete detection of serious adverse events for pharmacovigilance purposes, more complete detection of trial endpoints. It's a time and cost-efficient way of identifying serious adverse events and endpoints, and it concentrates the research staff effort on investigating the important events. It's also a relatively unbiased reporting source, and while normally studies rely on reporting from staff or patients, it's known that that's usually incomplete. Some of the challenges is you will miss events if they occur out with your record linkage area, so if someone has a heart attack in Spain, you will not pick that up. There can be problems with coding and miscoding. There's minimal information available to the study team when you see the codes and you have to try and interpret what the diagnosis might be. And it may limit the ability to assign causality or report pharmacovigilance properly. And it does usually require clinician involvement to try and interpret the diagnoses and assign causality. Sometimes secondary diagnoses are listed, so the patient may have gone in with one thing, but it also lists ischemic heart disease, and it's not clear if that was the reason for the admission or not, and may require further investigation. There's also the challenges of matching up reports received from record linkage with what the patient has already told you or what staff have already told you. Hospital transfers, each transfer creates another record, so it can be hard to link everything together. And there's a huge increase in staff workload at the time of each record linkage because, as you can imagine, we get thousands of events to look through at one time. There can be significant time delays. Some of the databases don't get filled for a number of months. And then what we find to be the most difficult thing is actually just gaining approvals for the release of the data in the first place. So we have to work with several different record linkage systems. I'm pleased to say that the Scottish one seems to work very well, but we have quite significant problems in some of the other areas with just getting approvals and constantly changing rules and regulations. So moving on, another study that we're currently running is the febuxostat versus allopurinol streamlined trial, the FAST study. This is looking at the cardiovascular safety of febuxostat versus allopurinol in patients with symptomatic hyperuricemia or gout. It was requested by the European Medicines Agency as a post-licensing safety study, and it's being run in the UK, Denmark and Sweden. And we have more than 6,000 patients taking part. And in this trial, one of the ways we tried to streamline things was to set up our own registered pharmacy premises to send patients directly their medication. Again, it uses record linkage for events, and this trial is currently finishing at the end of this month for the patient involvement, and we expect to get the results out in spring next year. So we set up a clinical trials pharmacy in the University of Dundee. We do direct distribution of the investigational medicinal product and the gout flare prophylaxis that we provided at the start. And it's delivered by post directly to patients' homes through their letterbox. And we've mailed out medication to more than 6,000 patients. The only country we're not allowed to do this in is Sweden, where we've had to go via a local pharmacy chain, and they then distribute it again to the patient. So we have sent out probably more than 200,000 medication packs now, and recently we're averaging about 800 packs a week. So it's been a really efficient system for us. Did you know that there's actually a European standard for letterboxes? The UK chose not to adopt it, unfortunately. But if you want to post things to Denmark, you get on fine. But in the UK, we have lots of funny-shaped letterboxes. So we actually did a bit of testing before we even started sending out medication. We posted out sample packs to staff to see how difficult it would be to get them delivered. And we had to refine and improve the packaging we'd planned until we got it right. And now the boxes fit nicely through the letterbox, just like a box of Hotel Chocolat, if any of you enjoy that. And we actually have a very good system set up. So what do patients think of that? They're generally very positive about it. It's very convenient for them. They don't have to come to a site to pick up their medication. They don't even have to go to a pharmacy. It's easy for them, but they don't like returning the excess drug, which we've asked them to do. And we did that partly to check their adherence. They don't like the waste, particularly in Scotland. The Scottish patients don't like wasting anything. And they don't like the inconvenience of having to post it back. But we think that this IMP supply system has really helped patient retention in this trial and helps us as well to keep patient contact details up to date. So we think it's been a big success and we will use it again. So, like every good trial, another trial comes along while you're still doing your trial to spoil everything. So this was the Kier Study. This was an FDA study in a higher risk population, so patients who already had cardiovascular disease. And this came out in 2018. And what it showed was that although there was no difference in the primary endpoint, which was cardiovascular death, MI, stroke, unstable angina with urgent revasc, there was a significant increase in cardiovascular death and all-cause death in the patients taking febuxostat. So obviously this made us carefully look at this data and think about the implications for our trial that was ongoing at the time. So the EMA and the MHRA later on issued new safety advice regarding febuxostat, and they advised people not to give it to people who had cardiovascular disease unless there was no alternative. We got quite a lot of queries, as you might imagine, particularly from GPs, because they received this letter from the MHRA. We had various discussions with our data monitoring committee, our trial steering committee, the sponsor, and the trial team. And these discussions had obviously been going on since the paper came out at the start. The MHRA contacted us and asked us to reassess our risk-benefit balance of the trial. And we were able to supply confidential information from the data monitoring committee to the MHRA, and they concluded that the trial could continue unchanged. So it was a secondary outcome in the Kier study. 57% of people in the Kier study stopped their study medication, and 45% of them were lost to follow-up. So there were significant issues, and it makes it quite hard to interpret. And we think that the European study is still really important in answering the question of which drug is safer in terms of cardiovascular risk. So we've sent out letters to patients, GPs, and investigators. And as I mentioned, the patient involvement will finish anyway this month. And then we're currently supplying a post-study supply of medication for 6 months to all patients. Another study that we're doing is the Allopurinol and cardiovascular outcomes in patients with ischemic heart disease study, ALL-HEART. This is funded by the NIHR HTA. And we're asking whether allopurinol improves cardiovascular outcomes in patients with ischemic heart disease. And again, we have nearly 6,000 patients with IHD randomized to receive either allopurinol added to usual care or to continue their usual care. It's a probe design, and the primary endpoint is a composite outcome of MI, stroke, or cardiovascular death. So this is funded to 2.7 million plus 800,000 of NHS costs. It's got remote follow-up largely, and again, we use record linkage data from the national Scottish and English systems to follow up the patients. The results of this trial are expected in 2021, and you can see that we've recruited from 424 different primary care practices across the UK. We did this trial by recruiting, try to get the laser pointer to work, by recruiting in primary care, so nurses went out to practices. We only had about 4 study nurses employed. The rest was done by network nurses across the UK, or practice nurses. We screened patients in a single visit. They were consented, screened, and if suitable, randomized. And then they were put in the allopurinol arm or the usual care arm. If they were given allopurinol, they came back in 6 weeks for 1 additional visit to have some blood tests taken and check safety. And after that, they go into a remote follow-up, so they don't have any further actual study visits. They're either done online by phone or by mail, and they have an annual questionnaire, partly to check adherence and partly to check for side effects and quality of life, and also health economic issues. So recruitment, we plan to recruit about half the patients in Scotland and half in England. As you can see, we ended up with slightly more in England, so it's about 60-40. And you can also see that at the very end, the recruitment shot up. So it seems to me that the best way to increase randomisation to your trial is to announce that you're closing randomisation. And then it's very hard to turn it off in time. And we had to justify going over by a few hundred patients at the very end because we felt that patients who already had been invited and had an appointment should be allowed to come through. And again, this was happening at 424 practices across the UK. However, we were allowed to keep these patients in the trial, and they're now all in follow-up. We randomised an average age of 72 in this trial, and about 75% of the patients were male. We see this in a lot of cardiovascular trials, that it's predominantly males who take part. They had a history of angina, MI, or other ischemic heart disease, and we also found that about 20% of them had diabetes at the time of screening, so they're a significant cardiovascular risk population, and we should know the results soon of whether allopurinol does help these patients or not. So the annual follow-up is ongoing. We have record linkages. As I say, in Scotland, we have no problems with these, and we've had 4. In England, we've only had 2 so far. There have been various delays in contracting and getting data. And then we go back to the original case records to pull out information about the event to see if it really was a heart attack or a stroke, because the coding on the record linkage is probably not detailed enough to be sure. And then an independent adjudication committee looks at these events, and they are blinded to treatment allocation in the trial. So we've had patients involved in the All Heart study right from before it was funded. We got their opinions on the study design, the acceptability of the planned randomisation, how they would see the study visits being very remote, and the remote follow-up. And we had 2 patients who joined the steering committee right from the beginning. They're full independent members. Originally, we recruited them from the Angus Cardiac Group. They've got a similar demographic to participants in the study, and they've got either knowledge or personal experience of heart disease. And they've also, both of them, had some experience of sitting on other health-related committees. They've really had invaluable input throughout. They've attended the meetings and participated fully. They've reviewed our planned amendments. They've had input into various study documents. And they give us good feedback, such as just make your sentences shorter. I mean, it's simple things that patients will tell you that don't always appear obvious to the trial team. They also said we should emphasise the positive aspects of our trials and not always focus on the negative and the possible risks involved with taking part in a trial. They're also amazed at the amount of bureaucracy that we have to handle to do any clinical trial. They were completely unaware of what goes on in the background before they participated in this group. So they've really provided us with the patient perspective throughout. So moving on to a fully online study that we're doing in Dundee, the TIME study. This is treatment in the morning versus the evening for patients with hypertension. So we're asking, does taking your therapy in the evening result in improved cardiovascular outcomes compared with morning dosing? Again, it's a probe design. The primary outcome is MI, stroke, or vascular death. And we have more than 21,000 participants in this trial. The grant for this was £1 million from the British Heart Foundation. So a huge number of patients for that size of grant. So the patients sign up online, they're randomised, they then go into an email follow-up, and then record linkage for outcomes in the 4 countries in the UK. They get email prompts every 3 months to fill in follow-up questionnaires. They are asked closed questions, and we've developed the questions with participant feedback. Recruitment, we did a lot of the recruitment in a more traditional way via letters going out from GP practices, about 16,000. We also tried recruiting from the UK Biobank, and again, we managed to get more than 3,000 patients from that route. We tried secondary care clinics and various other methods, but the most traditional way of going via GPs was the best way at that time. I think that as we go forwards, we can optimise our methods of recruitment and try and do more of the recruitment in more efficient ways in future. So the advantages to this trial, it's not geographically limited, so people who live up in the islands and so on can take part, whereas normally they would be excluded from research potentially. There's no need for any clinic appointments, and we can centrally coordinate everything from Dundee. It's faster, it's much less expensive, it is easier for patients, they don't have to travel to any visits, and we've been able to quite easily incorporate 4 different sub-studies looking at home blood pressure using patients' own blood pressure monitors, sleep quality, cognitive function, and mood. The participant feedback on this type of trial, in the time study, they've been generally positive. 5% of them said they would have preferred someone to explain the study to them by telephone rather than just reading the online information. Only 3% reported they would have preferred to meet with someone from the study. And 86% said they liked the fact the study was online. The reasons given for participation in the time study were predominantly to help others, altruism, or interest in medical research. Challenges, well, it sounds easy doing a study online, doesn't it? However, you get a lot of phone calls and emails to deal with. So when we were recruiting, we had more than 1,000 calls and emails during 4 weeks at the peak of recruitment, and 118 in one day. Now, even with a small team of staff, it's quite hard to handle all of that, and some of them require medical input or nursing input. And we also realised that we get about 47% of any email inquiries come in outside office hours. So again, we've got to cover the fact that there may not be an immediate response to some patients. And people will send emails saying, I'm feeling dizzy, I don't know what to do, what should I do? And that would normally require a fairly quick response saying, go to your GP or get some advice locally. And then email bounce backs are always a problem when people change their email address and we lose touch, at least temporarily, with them. So the 23rd of October this year started like any other day, really. We went into work, we went into a meeting, we had a cup of coffee, and then somebody knocked on the door and said, we're getting quite a lot of phone calls this morning. And the news had come out that day that a big Spanish study had reported that blood pressure pills work better at bedtime. So like I said before, when you're doing your trial, there's always another trial that comes along before you finish your one that spoils your day. So they kept coming, the Guardian saying blood pressure drugs work far better if taken at night. Take blood pressure pills at bedtime to reduce heart risk. Why taking blood pressure tablets at bedtime could prevent an early death. So this study reported, and everyone was reading this as they woke up that morning on the news and hearing it on the radio. And we got about 850 emails and a lot of telephone calls just from this media. So the Hygia chronotherapy trial by Hermida and his team was published in the European Heart Journal. It again looked at bedtime versus morning dosing of antihypertensives, and they had 19,000 hypertensive patients in the trial. The median follow-up was 6 years and the minimum follow-up 1 year. They gave these patients 48-hour ambulatory blood pressure monitors at least annually. Has anyone in the audience ever had an ABPM done? In this country, we normally do 24-hour ones. Did you enjoy it? No, okay. Most people don't enjoy it. I think 48 hours is quite long as well. I'd be surprised if many patients here tolerated that. They had 1,752 patients with their primary outcome, and their hazard ratio was 0.55, so they had a 45% reduction in their primary outcome just by taking your tablets at night instead of in the morning. And the cardiovascular death rate, again, was 56% reduction in cardiovascular death. So it seemed like a really large effect for such a small change potentially. It wasn't a change in medication, just in timing. The office and awake blood pressures in the study were reported to be very similar in the 2 arms of therapy. The asleep mean systolic blood pressure was slightly higher in the group dosed in the morning compared to the nighttime group, and the asleep mean diastolic blood pressure again was just slightly higher in the people dosed in the morning than at nighttime. There was quite a lot of discussion on social media about this, and if you look at the graph in the centre here, that's where the Hygia study comes out. This is change in blood pressure against the odds ratio of the events and cardiovascular mortality. And Hygia comes out way down here, so quite a huge effect size for the amount of blood pressure difference between the arms. So it remains to be seen whether this can be replicated. And there's various comments, it's probably too small for you to read, but just pulling out a few things. There's some concern about the transparency of some of the reporting of some of the angles of this study, and I think people are addressing that further in the scientific literature at the moment. So no patients in the study were reported as lost to follow-up, although 84 patients didn't complete the minimum of 1 year follow-up. There was a huge effect size. This was a primary prevention population only, so they didn't put people in who had cardiovascular disease. There was only a modest drop in nighttime blood pressure, and there were various adjustments made to the data, even though it was a randomized study. They didn't present the unadjusted hazard ratio, but it would have been interesting to have seen that. And we wondered why the trial hadn't been stopped early if the effect was so large. Didn't mention whether they had a data monitoring committee or not. So really, our conclusion on that one is that it probably does need replication. So the time study, as I say, is ongoing, and the BedMed study in Canada is also ongoing on this subject. But the effects it had on the time study, we had a huge increase in trial-related patient queries, as you might imagine. We know that at least 1,000 of the time patients switched their treatment time based on the media reports, which is unsurprising. If you heard that on the news, you might do the same. Many we know switched back later after a more informed discussion of what the pros and cons of that trial might be. And there were emails to all participants that we had to send out very quickly when the media stories broke, just to handle all the queries and inform patients of what had come out. We also sent a further email update the following week after we'd had a formal response from our own data monitoring committee about the future of the time study, because we did have to consider, should we continue our study if there's such a large effect in another similar study. So we don't yet know the full extent of the media effect until all our patients complete their next follow-up, but our data monitoring committee strongly recommended that we continue the time study as planned. So although we can't know the results yet, that gives us an indicator that they probably haven't seen the same as the Spanish study. So we're doing other things, just briefly. We're doing the evidence study. Can cluster randomization of prescribing policy be used to efficiently generate safety and effectiveness data within the NHS? So we know that NHS drug switching happens all the time. It's usually done for reasons of cost or availability of medications. And we decided to look at indapamide versus bendroflumethiazide. So when the BIHS and NICE guideline for hypertension came out in 2011, it was said that we should use indapamide or chlorthalidone in preference to bendroflumethiazide. But the practice in the UK continues that most GPs are actually using bendroflumethiazide. There's no great direct comparisons of the two drugs. There are some cost implications because indapamide is slightly more expensive, and there have been supply issues at times with indapamide. So the evidence study looks at whether indapamide is really better than bendroflumethiazide at preventing the major cardiovascular consequences of hypertension, and we're looking at those endpoints. We've completed a pilot in 30 Scottish primary care practices so far, which has involved about 7,000 patients. Only about less than 3% of patients have actually opted out of taking part in this study. In most trials, we ask for opt-in consent, but because this is a policy change in the practice, we were allowed to do this as a non-CTIMP in terms of a policy change and allow patients the option to opt out if they didn't wish to go ahead with a switch. And we're hoping to expand this and do it in a greater number of practices in the near future. We're also piloting remote systems for taking bloods in the community, so the iStat system where a little cartridge is put into a machine and you can test a patient's bloods on the spot. And we're also piloting online cognitive testing and hoping to use that in a large trial in the future. So moving on to the study that Tom mentioned, there's an IMI project looking at remote decentralised clinical trials. So Dundee is leading the first work package of this to look at what's out there at the moment and what's being done at present and what current case studies are ongoing using remote ways of doing clinical trials. So it's an international consortium. There's 30 academic, industry and SME partners, and it's a 5-year programme that we just started in September. So this is based on some US work that's looked at decentralised clinical trials, and the CTTI have made some recommendations about the way that we could improve, so faster recruitment, improved retention in trials, greater control, convenience and comfort for participants, and also increasing participant diversity. So we don't have to have an all or nothing approach here. So some trials lend themselves very well, such as the TIME study, to being fully remote. Others can be partly remote, so perhaps the patient needs to have some scans in a hospital or something, maybe in an oncology trial. They may even have to attend a hospital to have a delivery of an IMP, although increasingly that can be done at home. And there's a lot of trials could lend themselves to a more hybrid approach where we do as many aspects as we can remotely, but still have some study visits centrally. This was one of the first published studies that used a remote process. This was a Pfizer trial, and it looked at tolterodine in patients with overactive bladder. And the thing to note from this was that although 20,000 patients viewed the study introduction webpage, there was a huge fall-off at every stage, really, in how many patients continued. So by the time they got down to being eligible, there was only 1,500 patients from that initial 20,000. And if you follow it down, in the end, only 18 patients actually successfully completed the trial from the 20,000 patients they started with. So, as I say, this was an earlier study published in 2014, and I think there's huge lessons to be learned about how we can optimize recruitment and retention in online studies. The good thing about this is a lot of that drop-off occurs before you've had lots of staff involvement in terms of meetings with patients, so you're not wasting the patient's time if they're ineligible, and you're not wasting staff time if patients are ineligible. But I think we have got a lot to learn here. Another study that came out recently in the New England Journal of Medicine, this was an observational study, but again, there's some lessons to learn from it. So they used a smartwatch to try and pick up atrial fibrillation in members of the public, really. So people had their own Apple Watch and their own smartphone, and they agreed to take part by clicking on their phone. It was observational, and they managed to recruit a large number of participants. So it was 420,000 participants were recruited over 9 months, and this was a 100% virtual trial. So these are the devices that were used: Apple iPhone and a watch, and then participants where they had an abnormal rhythm picked up on the watch were sent an ECG monitor patch, and that was asked to be applied by the patient and then sent back. The pulse was monitored through photoplethysmography, and if there was an abnormality found, so if they had an irregular pulse picked up, they were then on to the first video visit with a study doctor. If nothing abnormal was found, they just went on to the end of study questionnaires. So if the e-patch was sent out and then it was returned, they either found the same arrhythmia or atrial fibrillation, and then they recommended that the patient contacted their healthcare provider for treatment. If there was no abnormality found, then they just proceeded with usual care, and then they completed an end-of-study questionnaire. So, as might be expected in the group who got a notification that their pulse was abnormal, lots more patients, a higher percentage, had atrial fibrillation, stroke, TIA, heart failure, MI, and major bleeding. And there were more people put on warfarin or anticoagulants, as you might expect. And you won't be able to read this, but I've blown it up on the next slide. But from the 419,000 that started, about 2,000 got the irregular pulse notification. But again, it went down to 200 patients by the end who completed the end-of-study survey. So there was drop-off at every stage. People had to put themselves forward for the first study visit. They then had to have the ECG patch shipped. They had to be able to return one that had worked and was able to be analysed. Then the second study visit, and then complete the end-of-study survey. So, interestingly, about 174 patients, although they were asked not to take part if they were known to have previous AF or flutter, still took part, and they were then excluded because that was found out when they had their first study visit, which was remote by video, as I said. And then there were various problems with quality issues, putting the patch on or returning it too late. And as you'd expect, again, there's a lot of lessons to learn from doing this remote way, because it's not the usual way of a patient coming to study visits with a nurse, where they have quite close support throughout. And this is a press release just from about a month ago. Companies are increasingly starting to do their studies fully remote or partially remote. And this is a Janssen study, chief heart failure, and they're looking at canagliflozin in patients who either have diabetes or don't, and this is going to be a fully remote study. So coming back to why do patients take part in clinical trials? Sometimes it's to get access to a new treatment or technology. Sometimes it's because they like the extra medical attention from a healthcare team. Generally, it's altruism and a wish to help medical research or to help others in the future with their condition. It's definitely not because they like queuing for hospital car parks, and it's definitely not because they want to have to take lots and lots of time off work during the day to attend study visits. So what we need to improve going forward is the recruitment, so the speed and the generalisability of the population we recruit. Partly we could recruit through using centralised NHS records, if that's allowed, to do with data access and privacy. Online recruitment is possible, but there are risks that you pick up the wrong patients or you don't reach the ones you want to reach. Patient experience of enrolment and participation, it has to be a good experience. If you use websites you don't like, you're probably not going to bother using them again. So we have to make things easy for patients and rewarding for patients, preferably feeding back some of their own data to them so they feel that they're getting a value from taking part. The quality of data collection has to be good, and there are different problems with running remote trials, and we're still learning all the challenges and those solutions. So I think that remote decentralised clinical trials are the future. We need to work on improving the ease and the quality of the experience of trial participation. We need to work on improving the efficiency for the trialists and the sponsors and funders. And we also need to optimise the use of technology in trials. Just because we have technology doesn't mean we should always use it. We have to use it in a good way for the group of patients we want to reach. And we have to keep patients at the heart of the process throughout. So if you can collect data from a children's study by putting an electronic teddy bear in that they can press buttons on, why not? So I'd like to acknowledge clinical trials are a huge team effort. We can't do them alone. So my colleagues in Memo Research in Dundee, collaborators all over the UK and Europe and worldwide, the patients who've taken part in our trials, and the funders. Thank you very much.
Excellent, Isla. You made it through without fainting, or?
Thank you.
And bang on time as well. We've got 5 minutes for questions from the audience. If you'd like to raise your hand if you've got a question. There are lots of issues. You can see that universities are about to go on holidays for the festive period, but of course, trials don't take holidays, and patients have serious adverse events that occur all the time. So that's one of our challenges that we face when running large clinical trials like this. We get no holidays. I think there's a question down here. Is that Jeff?
Thanks, Isla. Beautifully simple. David Grahame Smith would have been delighted to hear that. I'm always in awe of people who do these enormous trials with all the thousands of patients multi-centre and all the organisational difficulties that you described. It's fantastic. And I was interested that you are measuring or trying to measure adherence because it's so often neglected and is obviously so important. And in the one case I think you mentioned you were using return of tablets. I wonder if you're using other more modern methods with electronic adherence measures.
So there are lots of ways of measuring adherence. So there are smart dispensing containers, for example. There's smart capsule dispensers where you push it out and it records that the patient's done that. You still don't know if they've swallowed it. You can put tracers and things into patients. It's probably a bit extreme, but there's no perfect way to measure adherence in clinical trials. And with the online methodology, it becomes an even bigger issue because you have much less direct contact with patients. So I think, again, that's an area that we need to learn more about for remote trials, but there's no good way at the moment of doing it.
What about urinary metabolites, Isla? Would you?
You can. I mean, in hypertensive field, we've been measuring urinary metabolites, but again, you know, you're basing that on what the patient's told you they're taking, you're measuring something, then you still have to do something with the result when you get it. So you then have to have a difficult conversation with the patient, and it can be very difficult. But within trials, I think you also have an obligation to make sure that patients are adhering if you can measure it in any way.
Hi, it's great to see this area moving forward. I had a question about the data collection is also related to actually mining the data afterwards. So that depends on, for example, one thing, the data flowing into major databases, so it can be data mined. And the other thing, whilst clinicaltrials.gov has its imperfections, are all these virtual trials being recorded there?
So yes, they should be pre-registered on clinicaltrials.gov like everything else. And in terms of data mining, I mean, you do have to be careful. You're not just collecting data just because you can. There's a huge potential to collect, 24 hours worth of data on everyone for everything. You have to decide what's useful and sensible for your trial, and also what's possible for the participant, because just because you can collect it, it may be that the patient doesn't want to wear a wearable vest all the time. It might be okay just to wear it for a week and then stop wearing it. And you really have to decide what's sensible, how much data do you need to get your outcomes.
There's a question down here. Yes.
Yeah, I wanted to ask, obviously when you're doing these remote trials, you are taking patients essentially by the word, and you're relying on the way of explaining correctly what's happening to them and how they're adhering. How do you account for misreporting specifically when you're doing remote trials?
So I think that's a very good question. So particularly ensuring that patients have the diagnosis you think they have when they enter the trial. So there are ways around that. You can get them to agree for you to contact one of their healthcare providers to verify it. You can have some kind of interview with the patient to try and establish, but again, it's not going to be perfect. So, I mean, we have that problem anyway in the trials we do in a more traditional way, but I think you're opening up the potential that people could wish to take part and therefore give false information to be able to take part. You've also got issues of verifying it's actually that participant. So, yeah, it all needs working on.
Thank you.
Question up here.
Thank you, Isla. Wonderful work. I have two questions. In the record linkage part of your work, how do you deal with the eternal problems of missing information and misclassifications in these databases? And my second question, you certainly have a better record linkage system in Scotland compared to England. So what data sources you use in England? Because CPRD and THIN cover only 7% of the people. How do you get national coverage of record linkage in England?
So in England, we use NHS Digital, who do potentially have all the hospitalisations and deaths. We just find that getting the data takes a long time, and particularly getting the approvals, and we do have to keep changing contracts and updating agreements, and that's been the biggest delay. In terms of coding issues, we're quite inclusive when we review a record linkage, in that we include anything that might be an MI or a stroke and look further for it. But we can't get by something that's completely misclassified. So if it's been wrongly coded at source and we don't know it's wrongly coded, we may miss something. But we do always go to original records or GP letters to try and verify anything that we pick up from record linkage. There are some teams who say that we should just be using record linkage alone. I don't think we're there yet. I think for MI that might be possible, but for everything else, I think there's too much grey in amongst the codes that we get.
Well, ladies and gentlemen, I think you'll agree that was a fantastic lecture. By the future of medicine is standing there. That's what we're going to be doing from now on. And if we don't do that, some drugs have cost 1.3 billion just to get on the market. Then is it a surprise they cost so much? We have to do these trials more efficiently, to a higher standard, with better evidence if we can. Otherwise, we're going to have a bankrupt pharmaceutical industry, or we're going to have to pay more tax. So, Isla, thank you very much. That was a fantastic lecture, and could you please give her a round of applause?
Thank you.